Hi, my name is Jeff Lancet. I'm at the Moffitt Cancer Center in Tampa, Florida. And we often get asked about the drug CPX-351, also known as Vyxeos or liposomal, daunorubicin, and cytarabine. Questions about the quality and the depth of the remissions that are obtained with this compound. Now, CPX-351, as most people know, has been demonstrated to result in superior overall survival amongst patients with secondary or therapy-related AML compared to traditional 7+3 induction chemotherapy. That's been established. But the precise reason for this benefit is still somewhat unclear. And a question often comes up, well, do you achieve a better or a deeper remission with CPX as an explanation for why an overall survival was observed in the clinical trials? And to date, we don't have the answer to that question. Unfortunately, we were not able to study post-remission or measurable residual disease amongst patients in the original phase 3 trial. So we don't have an answer as to the depth of the remission amongst CPX-351 treated patients. What I will point out to you is that amongst all the different molecular subtypes that were studied within the context of this trial, they all seem to benefit from CPX-351 compared to 7+3, with the exception of patients that carry the TP53 mutation. So the survival advantage seemed to be independent of the molecular status, except for the case of TP53. Another interesting observation presented at this year's ASH meeting by Dr. Jeffrey and colleagues was that amongst the transplanted patients on the phase 3 clinical trial of CPX versus 7+3, as you may recall, the transplant outcomes were superior amongst the CPX treated patients. The reasons for which were not completely clear. And again, relates to the question of whether maybe a deeper remission was being achieved with CPX that would explain the better survival after transplant. So although we don't have that data specifically to share at this point, we do recognize from that analysis done by Dr. Wee that one major reason for improved outcomes after transplant was a reduced treatment-related mortality effect amongst patients who had received CPX-351 and then went to transplant, compared to 7+3 followed by transplant. So it's possible that the survival benefit that was observed after transplant was largely related to a better patient condition and overall clinical status going into the transplant that may have led to a reduced mortality during transplant or shortly after transplant to explain the benefit. But we still have work to do to better understand the molecular features of AML after CPX-351 and how that drug in particular may impact the natural history of the disease in its own specific way.